Everyone. My name is Maria Castano, and I'm excited to open the afternoon session of the Hamilton Symposium today. And so today I'm going to talk to you about some of what I've been doing for my PhD at the University of Rochester. So vivid coloration evolved over time across many different lineages and is one of the most conspicuous traits that varies among organisms. Because it's visible, quantifiable, and often linked directly to fitness, it has been a powerful model trait for studying evolution and link genotype to phenotype. So using SNP data and genome-wide association studies, we've made significant progress in understanding the genetic architecture of pigment-based coloration. For example, melanin-based coloration is often linked to well-characterized genes like MC1R or ACIP, and these associations are often simple, sometimes involving single genes with large effects. But since carotenoids are obtained from the diet and have to undergo metabolic processing, their genetic architecture is often more complex. So some known genes include CYP2G9T, BDH1L, and TTC39B, associated with red versus yellow traits in many vertebrates, or the famous gene BCO2, associated with white versus yellow color in chicken legs and in bird feathers, plus a few others that have been identified in fish, lizards, and other organisms. So our knowledge on the genetic architecture of color traits is largely based on short reads and SNPs, but as is becoming increasingly evident, and we've seen previously in this symposium, there may be other sources of genetic variation that can contribute to these phenotypes. So to study this, for my PhD, I've been working with the Flame-rumped Tanager subspecies complex, which are found along altitudinal gradients in Colombia. So they have very dramatic variation in the carotenoid-based plumage color of the rump. The lemon-rumped tanager is found at low elevations in the coastal plains of the Pacific. And in the other side of this mountain, we have the flame-rumped tanager, which are found at mid elevations and are endemic to the Cauca Valley of Colombia. And so wherever they meet along low mountain passes in the mountains, they hybridize extensively, and they are the living proof that yellow plus red equals orange. So more technically, interbreeding between subspecies creates this remarkable variation in the rump plumage in a relatively narrow area where the species hybridize. So as you can see from this phylogeny, all the other members of the Ramphochelys genus are red. And from another study, we know that in this clade of birds, red is ancestral and yellow is derived. And in fact, our lemon-rumped tanager is the only yellow male in the whole genus. So in previous work for my PhD, we sampled 3 independent hybrid zones in the low passes of the Western Andes of Colombia, and we found that genomic ancestry predicts rump plumage color almost perfectly, as you can see in these plots of hybrid index in the x-axis versus rump color on the y, which show a strong correlation in all 3 transects. So we were interested in finding which genes may be responsible for these changes in phenotype that we observe. So for this, we first sent 161 birds for whole genome sequencing at 20x, from which we had 93 hybrids. And we did 2 traditional genome-wide association studies, one with k-mers here on top, which doesn't require a reference, and one with SNPs in the bottom, which was done based on mapping the short reads to a reference genome for the yellow female that I assembled as part of my first chapter. And so both show similar association signals, with the stronger ones located in chromosome 3, 8, and 22. In chromosome 3 and 8, we have the genes BDH1L and the famous carotenoid ketylation gene CYP2G19, which together convert yellow dietary carotenoids into red ketocarotenoids in many vertebrates. And this reaction is further enhanced by TTC39B, which was in a near significant peak in the Z chromosome. But you may be wondering, what is in this massive peak in chromosome 22? Well, the association occurs around the gene REDSAT. So this REDSAT gene has been linked to the pathway for carotenoid metabolism in the bird retina, where it degrades yellow dietary carotenoids into less pigmented forms, which after deposition in the SWS2 cone, they allow birds to fine-tune and better discriminate colors, particularly in the short wavelength portions of the light spectrum, like yellows through blue. And only our yellow feathers reflect in those shorter wavelengths of light, and they also have a peak in the UV. So this gene has been studied in the retina, but we thought it may be catalyzing a reaction in the feathers that just had not been described yet. And so, in addition, we performed a differential gene expression analysis using RNA I extracted from red, orange, and yellow emerging feather follicles and tested for genes whose expression changes with changes in the color of the rump. And what was very cool was that of the 37 differentially expressed genes, red SAD showed the highest fold change, so 14-fold between yellow and red feathers. So in the plot to your right, you can see expression decreases with increasing Q. So in other words, the redder the bird, the lower expression levels, whereas yellow birds have higher expression of this gene, which was very exciting. Though, as you may have noticed, the associated SNPs and Kmers span to around 700 kb of the first megabyte of the chromosome, which made us think that this region was maybe linked by some type of structural rearrangement. So we sent 9 more samples for long-read PacBio hi-fi sequencing to test whether structural variants were playing a role in shaping this color phenotype. So in total, we had 10 birds, 3 yellow, 4 hybrids with a range of oranges, and 3 red. So we assembled 20 haplotypes in total, which were all highly complete with more than 95% BUSCO scores. With those 20 haplotypes, we built a pangenome for the species, which not only allow you to get the usual substitutions or simple structural variants like short insertions or deletions, but they allow us to capture variation at a much bigger level where things like big inversions, insertions, and duplications are included in the graph that you then use to map your reads. And so you can go beyond SNPs and test for how structural variants contribute to species divergence instead of biasing your analysis to a single reference. So when we looked at our region of interest in chromosome 22, we saw a bubble in the graph that contained a copy number variant at the red SAD locus, represented in dark blue in this schematic. So our analysis showed that red birds carry 2 copies of red SAD per haplotype, so 4 per diploid genome. Yellow birds carry 3 copies per haplotype, so 6 per diploid genome. And hybrids carry 5 to 6 copies per diploid genome. So the CNV is embedded within a broader duplicated region that includes the gene red SAD and a housekeeping gene LMOD3 here in light blue. So in the reference yellow assembly, the red SAD gene is 6 kb long and has 3 copies, all flanked upstream by a copy of LMOD3. The lines on top are the annotated repeats, and as you can see, the red SAD copies are also flanked by transposable elements, specifically lines, and LTRs, which made us make the total duplicated segment of about 19 kb long. So this suggested to us that this may be a case of unequal crossing over mediated by transposable elements, which facilitated the tandem duplication of the 19 kb segment. So to validate the copy number variant, we mapped the hi-fi reads back to the yellow assembly, which has the 3 copies, and the depth of coverage confirmed the copy number assignment across individuals, with yellow birds showing consistently higher depth across the duplicated region, while red birds show a valley along that middle copy. Then, to reconstruct the evolutionary history of red SAD, we built a maximum likelihood gene tree, including all red SAD copies from both parental subspecies, along orthologous sequences from other birds that have good quality assemblies, like the zebra finch, birds with well-studied variation in carotenoid-based color, like the house finch and canaries, and closely related taxa in the Tanager family, including the sister genus to Ramphocilus, and they all have 1 copy of this gene. But when we look at the only other Ramphocilus species with a published assembly, the wild-like tanager, we saw that the duplication is present. And finally, as you know already, our subspecies have 2 copies for red birds and 3 for yellow birds. So it seems that the duplication occurred within Ramphocilus after divergence from the sister clade. But what was interesting is that within Ramphocilus, gene copies clustered by subspecies rather than by copy number, a pattern consistent with concerted evolution caused by ongoing gene conversion, homogenizing the copies within each subspecies. And so because gene copies are almost identical, when we mapped the short reads for the 160 individuals to the yellow reference, they mapped in similar proportions to each copy, which causes consistently lower coverage across red birds as reads are spread across the 3 copies, intermediate variable coverage in hybrids, and consistently higher coverage in yellow birds. So ultimately, we saw this correlation between plumage color hue and the mean coverage across the 3 copies, suggesting that the dosage-driven expression of red SAD is influencing plumage color. But you may be wondering, what role exactly does reds at play in the color differences. So to produce feather coloration, birds use carotenoids they obtain from the diet, mostly in yellow form, which can be deposited in the feathers unconverted. So they deposit the pigments the same way as they ingested. Or dietary carotenoids can be chelated by adding a ketone group to the molecule, which shifts the absorption maximum to longer wavelengths, causing a red shift in the pigment color. In contrast, pigments can be saturated by removing some of those double bonds from the chain, which shifts the absorption maximum towards shorter wavelengths, causing a shift towards the green, blue, and UV. So based on that knowledge, and the fact that in our GWAS we have peaks at both a ketolase and a saturase gene, we decided to characterize what pigments were present in our feathers and at what concentrations using HPLC. And so in this chromatogram, the top line is a red feather, the middle line is orange feather, and the bottom line is a yellow feather. And we found that the main dietary carotenoid in yellow feathers was lutein, and the metabolized ketocarotenoid in red feathers is alpha-doradic-xanthine. But there were two more peaks corresponding to saturated versions of these two pigments. And so if both yellow and red feathers have saturated pigments, why is red sat our top differentially expressed gene and the highest US peak instead of, say, CYP2J19 in terms of predicting plumage color? And for this, it is important to remember that saturation reactions progressively shift the absorbance to shorter wavelengths with increasing degrees of saturation because we think the answer comes back to the red sat dosage. So here we have the proportion of each type of carotenoid pigment in the feathers being by subspecies. And we clearly see that yellow birds have higher proportions of double and triple saturated carotenoids than hybrids and then red birds. And this grouping by subspecies also corresponds to the copy number of red sat. So the less copies of red sat you have, the less saturation reactions you can make, the less pigment shift to shorter wavelengths. So based on this, we propose a two-step metabolic pathway for color determination. In the first step, dietary lutein is converted to the ketocarotenoid alpha-doradic-xanthine, catalyzed by CYP2J19 and BDH1L, shifting plumage from yellow to red. And in the second step, both lutein in yellow birds and alpha-doradic-xanthine in red birds are saturated by red sat, producing two saturated pigment in parental red and yellow feathers. In hybrids, however, we hypothesize that the precise shade of orange depends on the joint genotype at both red sat and CYP2J19. For instance, if they inherit the red CYP2J19 allele with 5 red sat copies, they will have more saturated carotenoids, producing a lighter orange hue than parental flame-rumped birds. In contrast, if they inherit the yellow CYP2J19 allele, but only 5 red sat copies, the dietary carotenoids will be less saturated, retaining a warm and lutein-based yellow pigment compared to parental lemon-rumped birds. So with this results today, I showed you how a pangenome graph allowed us to discover a gene duplication driving color diversification in birds, which apparently have found many ways to transition between red and yellow plumage. But more importantly, our results revealed that while amazing insights have been gained over the past decades from traditional SNP studies, we may have been missing key structural variation that can drive significant phenotypic divergence. With that, I want to acknowledge all the people and societies that helped me conduct my work, and I'd be happy to take any questions if there's.
So we've got about a minute and a half. If anyone has a relatively quick question, please go ahead and raise your hand, and we will get a chance to call on you. All right, I guess with no other questions pending, I'm going to ask, what do you want to do next with this system?
Well, we are trying to time when this duplication arose and whether it has any selective advantages, because there's a lot of differences in the two environments that the tanagers inhabit. So one is the most humid place in the planet with a lot of vegetation and canopy cover, whereas the other one lives in a much disturbed ecosystem that is much drier as well. So we want to see if yellow actually has an adaptive advantage in those ecosystems where the lemon-rumped tanagers live.
All right, that sounds good. I am going to ask you to stop sharing your screen so Brad, our next speaker, is able to share his.
Okay.
And then we're going to wait the remaining 10 seconds before you want to start your
Sure.
next one. I will also direct Maria to note there is a question in the Q&A for you to answer as you have time. Brad, you can go ahead and start whenever you want.
Okay, thank you. Thanks for having me. So I want to introduce a framework for studying and think about selection, and it's really including the protein structure information into our analyses. And I'll show how that can reveal some patterns of selection that are hidden when you just approach genes as linear or just sites. Um, let's see. There we go. So I want to start with just two kind of facts. And one is from the evolutionary biology perspective that selection acts on phenotypes. The other fact is from molecular biology perspective that protein structure really determines the function and the phenotype of that gene. And so bridging these two, we see that selection is often targeting structurally organized kind of 3D features of proteins. And so we need kind of 3D methods to uncover these features or the selection of these features. Examples are protein-protein interfaces, overall stability of proteins, catalytic domains. Really anything about protein function is about 3D organized residues. Um, but we often think about selection at SNPs, codons themselves, or sliding window, linear sliding window through a gene. And really, we're missing a lot of the structural information about how proteins function and how selection acts upon them. So I'll show how you can simply add back the structural information before I show how I've used these analyses. So I'm sure lots are familiar with linear sliding windows, just sliding a window through the gene, capturing a central codon and its sequential neighbors. And a spatial sliding window is very similar, but you rank neighbors on their 3D proximity. So here's an example of one spatial window, the codons where this window comes from, and then you can slide that spatial window to a neighboring codon and grab a new set. And so if you think about linear windows, you simply subset the MSA based on the window at hand, and the spatial window is the same. Grabbing the codon columns that correspond to each spatial window. And then we coin a term, spatial haplotypes. You build a structure-informed subset of the multiple sequence alignment. And this gives you back this structural information. And I want to argue that really selection is acting on these 3D environments, not residues by themselves and not residues in sequential order, but actually the site where the protein function emerges, which is this 3D environment. And there are some other advantages also to structure-informed analyses in that some aspects of protein biology that really aren't accessible through linear methods are kind of naturally fall out of a structure-informed method. So one of those is multi-gene neighborhoods. So this is many genes coming together to make one surface, and you could imagine a receptor binding domain made of many genes. Essentially, selection acts on it as one unit, but it's coming from many different places in the genome. So with structure, these are easier to get to. And interfaces also can be treated as analytical units. So in pink is a protein, and colored here are the residues that contacted this protein. So each different color is a different protein chain in itself. So it's kind of a cross-chain interface. But you can ask questions like, is there balancing selection at this protein-protein interface? Is there conservation? So I really just wanted to first highlight this this structure-informed method of thinking about and analyzing selection. And I do want to highlight, I made a R package, but it's more just to say it's simple to use if you want to try these things for your own research. So maybe you have SNPs after GWAS, you'd like to understand where they map on this structure. You have population genetic data for a protein, you have sequence alignment data across taxa. Um, yeah, I made a package so it's very easy to get to these methods before I talk about how I use them. And with AlphaFold, really, there and PDB itself, there really is not so much a limiting factor on getting structural information. So it's a good time to think about adding structure to selection analyses. And yeah, I would like to make it easy for people to use. So I made it, you just have to know one function in R. If you have genetic diversity, you have MSA file, and you have a structure file. Getting to these spatial haplotypes is kind of automated from that. So, okay, I will now move on to things I've applied this method to. The first is vaccine design. Essentially, we want to look at evolutionary constraint on pathogen proteins to know where will it be good to target this, where can antibodies target this pathogen protein? One, so it has good population coverage, and two, maybe so it's hard for the virus or bacteria, anything really, to mutate around those antibodies. So hepatitis C is a good example because it's extremely genetically diverse, um, and there currently is no vaccine for it. And it's so diverse because it's always having to avoid the human immune system, memory antibodies. So it kind of wants to maximize its diversity, but there's still some constraints. Some surface patches lack diversity, they're conserved. And so the primary target is this E1, E2 protein. So we started with this, and we have 271 genomes. And the idea is we can scan the surface as if we're an antibody and characterize what's the conservation at each of these 15 residue surface patches. And the goal is to find some conserved epitope-sized patches on the viral surface or the protein surface. And so just an overview, we have the E1, E2 complex. We scan the surface in 15 residue patches. We build spatial haplotypes for each patch. And then there's a number of statistics you could apply to each of these spatial haplotypes, these MSA subsets. We went with block entropy, which we borrow from T-cell vaccine research, in where they look for conserved T-cell epitopes, which are peptides. But really, no one has done this for conserved antibody binding sites because antibodies always bind spatial regions. So you need a method to map back and forth between genetic diversity and spatial patches. And block entropy is Shannon entropy, but instead of doing it at single sites, you do it at the haplotype level, so over haplotype frequencies. So here's the results of this hepatitis C investigation. So in blue are conserved spatial regions, and in red are very diverse spatial regions. And there's a little fact here that the median surface patch is 3.3 bits. So it underscores why it's been hard to find a vaccine for hepatitis C, because one vaccine would have to raise antibodies that target essentially 13 versions of the surface. And this is common for lots of pathogens of high diversity. It's hard to find a single vaccine that could cover the population as a whole. I want to highlight this large blue region. We know why this region is conserved, and that it's, well, one copy of this E1, E2 molecule binds to another copy, and that makes a dimer, and this is the dimer interface. So across our 271 genomes, there's only one version of this surface. But antibodies can't target it. It's hidden from antibodies in that dimer. So if you flip to the exposed face, antibodies have access to this face, and you can kind of see the history of immune targeting here. So this is extremely diverse, and antibodies to one version of this spatial region won't cover pretty much any other or most of the other versions of this region the pathogen population has. But we do, and it's a very nice finding for us, we do find conserved regions on this exposed face. If you see two residues in blue, though the residue is blue, it really means that the entire surrounding region of this residue was very conserved. And same with this one up here. So 0.6 bits, 0.7 bits, it means more than 90% of the genomes we sampled have the same haplotype, the same spatial haplotype at this surface. So it'd be a great target, and it raises questions. Why is it conserved? the virus itself wants to kind of achieve this maximum diversity to avoid memory antibodies, but for some reason, and I don't have any I don't have any insights on what that reason is, but for some reason these patches are conserved. So it's good for two reasons. Um, Antibodies targeting this would cover a lot of the pathogen population. And then second, it was probably hard for the virus to add mutations in these regions to avoid a vaccine. So lots of my ongoing work is kind of continuing this work for other highly diverse pathogens that we lack vaccines for. And it's one example of adding structure information. Oh, the last thing to say, in the linear view, if you look at the linear sliding window around this residue, it seems diverse, but antibodies are actually binding the spatial region. So yeah, the spatial region here uncovered some hidden conservation. And I think I have 5 minutes. So the next one is more of a like a serendipitous kind of result. Really, there's this huge protein complex, and I wanted to show that the R package could handle such large proteins in a reasonable time. Um, it has some known interaction partners with human proteins, and we do the same idea, this block entropy analysis. We found a hotspot of diversity centered around residue E2, 119. But we kind of have a bit of a confusion because this residue is also part of this binding interface with the human protein. So we would think the binding interface should be conserved. You need to target the human protein, but we're finding the highest diversity on a residue or centered on a residue within that interface. Um, so we extract the interface directly to investigate, is it conserved, is it diverse? And to add one more fact to this, actually E2 itself, or 119 itself, doesn't have any changes. It's completely conserved, but the region around it is very diverse. So when we pull the interface, 23 out of the 35 residues are completely conserved, 32 are very low diversity, and some have two amino acid options. But essentially, it means the interface is conserved, the diversity is concentrated near the interface. So it's kind of this push and pull, this spatial partitioning of selection pressures. One, you would like to conserve the actual interface, but you also need to avoid antibodies. So it seems there's a pressure to add diversity, but that pressure, the balance changes once you get into the interface. Let's see. I think maybe in the interest of time, I will skip this. It's just people have now started to ask me to use this package to maybe contextualize some findings after GWAS on SNPs and things. But I would, it's not too much to say, sorry. So really, using these structure-informed methods, which we're at a good time to start using them, does capture a different aspect of protein biology that you won't see at the linear level, and it's probably a more realistic kind of field where selection is acting, these 3D environments of proteins. And so, yeah, for vaccine design, we can use pathogen evolution to actually tell us where good targets will be against them. And we were successful on hepatitis C in this example. And you can directly study interfaces, pathogen host interactions, or within this one organism interactions. And you can see that different aspects of the protein as a whole, different spatial regions, are under different selection pressures. And then the last, I just want to inspire, maybe I would like to see more papers use this. I think this is a very powerful approach, but the lack of structure historically kind of leaves it underutilized, unexplored. But now structures are available, and I spent a long time trying to make it very easy for other people to do such an approach. So yeah, hopefully in the future we see many more structure-informed analyses, and I look forward to these papers. I'd like to thank my advisor, Shishin, and the He Lab. I'd like to thank the funding and the platform to share at Evolution Conference. So thank you, and take some
Thank you.
questions.
We are not going to have enough time for you to have any questions. I'm sorry
Have
about
them in
that.
the chat. Thank
Um,
you.
so, Gabrielle, do you want to go ahead and share your screen? For people who do want to ask questions, I again direct you towards the Q&A. You have the ability to ask some questions there. But Gabrielle, you are ready to start whenever you want.
Awesome. Hello, everyone. I am Gabrielle Welsh. Thank you so much for coming to my talk today. Today, I want to tell you a story about how, when, and why new cricket songs are evolving and spreading in Hawaii. So, some of the most spectacular and diverse traits that we find in nature are the signals that animals use to communicate with one another. And these traits contribute to the origins and maintenance of biodiversity. Often, when we look at closely related species, the most obvious traits that differ are these sexual signals, even if they have widespread similarities in other traits. Sexual selection has even been called an engine of speciation. Yet our understanding of the earliest stages of sexual signal divergence is hampered by the inability to directly watch that process. Instead, we're often looking back in time, making comparisons between closely related species that already differ in their flashy displays. But what if we could watch the origins of novel signals in real time? What features of signalers, receivers, and evolutionary history would allow novel signals to become established? A great study system to investigate these questions is the Pacific field cricket, Teleogryllus oceanicus, which has experienced a rapid radiation of novel signals over just the last 7 to 8 years. That means we have had the opportunity to watch novel signal evolution happen in real time. These crickets originally evolved in Australia and island hopped throughout the Pacific, arriving most recently in Hawaii. And in Hawaii, they encounter a parasitoid fly, Ormia ocratia, introduced from North America. Importantly, these two species only encounter one another in Hawaii. The parasitoid fly is not found elsewhere in the cricket's range. This introduction of the cricket to Hawaii occurred sometime between 700 and 1600 years ago. And upon introduction, all of the male crickets made what we now call the ancestral or typical song. So male crickets produce songs using their wings to attract females for mating. So the picture here is an ancestral wing with all of the sound-producing structures highlighted in blue and green. Now, just 35 years ago, the parasitoid fly was introduced to Hawaii, and gravid female flies use that ancestral cricket song to locate their cricket hosts and kill them. As a result of this deadly interaction, a silent cricket morph evolved and spread 20 years ago. So silent males' wings lost not nearly all of their sound-producing structures, which I'm showing here. So when these males stridulate or rub their wings together, no sound is produced. More recently, at least 4 different novel sexual signals have evolved amongst Hawaiian males, each with different wing morphologies that generate different songs. And we call these purring, small wing, curly wing, and rattling. Here are spectrograms for the songs produced by each wing type, showing the dramatically different acoustic characteristics, from the loud, tonal, ancestral song on the far left to the novel wings on the right that produce quieter, more broadband songs that protect the crickets from that parasitoid fly, but still attract females for mating. And through longitudinal sampling, we are seeing that a lot of these novel morphs are becoming more common in Hawaii. There are a lot of interesting patterns that I could talk about in a snapshot like the one I'm showing here, but what I want to draw your attention to is that in 2020, when I started my dissertation, purring in salmon was dominant in the 4 of the populations that we regularly study, and rattling in red was the dominant morph in Hilo. So I'm going to focus on how these 2 novel morphs got a foothold in the rest of my talk. As I pointed out on the previous slide, these purring and rattling songs sound very different from that tonal, loud, ancestral song. And this begs the question of why do female crickets accept these novel song types? So I'm going to tell you 2 stories about rapid evolution in this system. The first asks an ultimate question and centers on how and why the novel song variation is able to stick around and spread within populations. The second addresses a more proximate question that centers on what underlies this variation at the molecular level. So first, how is this variation able to stick around and spread? To answer this, we need to know a bit more about female preferences and how they function. So signalers and receivers often exhibit tightly knit relationships, and this can make it difficult to imagine how novel signals evolve. For example, say on this preference function, the female cricket, shown on that y-axis, readily accepts ancestral males on the x-axis. But if a novel purring song evolves and is not perceivable or acceptable to the female, most females will reject these males, and this signal should be removed from a population. The same should happen with any novel signals that pop up. But if a female is accepting of a broad swath of male songs, indicated by this high and accepting preference function, this relaxed preference could allow novel signals to persist long enough for more direct preferences to later evolve. I tested this idea by investigating two ways that relaxed preferences might emerge: selection and plasticity. So first, selection for relaxed preferences may have either preceded or followed the evolution of these novel morphs. For instance, as these crickets colonized new islands in the Pacific, they experienced strong genetic bottlenecks, with few individuals making it to each consecutive island. Kaneshiro hypothesized that in small, recently founded island populations, very choosy females might never find an acceptable mate, leading to selection for females who are less picky. Of course, an alternative and/or additional mechanism is plasticity, stemming from experience and environment. And my experimental design encompasses both of these options. So the first step in a successful cricket mating is a female locating a singing male from afar. So I used phonotaxis trials where I placed each female at one end of an arena and measured her phonotaxis behavior in response to stimuli, including the novel purring and rattling songs. I did this using field-caught individuals in 6 populations, 2 in Australia, 2 in French Polynesia, and 2 in Hawaii. If Kaneshiro's effect is at play, island populations should be more responsive to these novel songs, and these patterns should hold in Common Garden. So I repeated the experiment in the lab under Common Garden conditions. If plasticity is important, we should find differences in female responsiveness between the lab and field, because these two conditions expose the animals to different environments. I'm first going to show you the field data with proportion of females that were phonotactic on the y-axis and the region, Australia, French Polynesia, and Hawaii, on the x. First, we found a significant interaction between region and song. Regions did not differ in the proportion of females that were phonotactic to silence in black, or to purring in salmon, but we often see low levels of responsiveness to purring because it's such a quiet signal. But when we look at the novel rattling song, shown in this deep reddish pink, we see that Hawaiian females were the most responsive to rattling, French Polynesian females intermediately so, and Australian females were not interested. We see the same pattern in response to the ancestral song in teal, consistent with females in Australia being very choosy. So what happened in Common Garden in the lab? Here, females exhibited higher responsiveness overall than they did in the field. And importantly, those differences among regions disappeared. Here I'm showing you the lab and field data side by side, so we can directly compare the height of the bars. And we clearly have differences between the two, indicative of important plasticity. So I've illustrated both evolutionary changes and plasticity that generate relaxed preferences. And this relaxation helps answer the question, how does this variation stick around and spread? Relaxed preferences allow novel signals to persist long enough for populations to perhaps climb new fitness peaks and perhaps for novel preferences themselves to eventually evolve. But what underlies this novel signal variation at the molecular level? And how can this system help us understand mechanisms of adaptation? So the purring and rattling males both produce greatly attenuated signals, but they do so with different morphological changes. Recall that crickets make sound by stridulation, or rubbing their wings together. So here is a traditional ancestral wing with all of the standard sound-producing structures labeled. Rattling wings look identical to ancestral wings, except that they have gaps in their file, where teeth should be but don't develop. And I'm showing some zoomed in images of this here. purring wings, on the other hand, have no mirror, that circular structure, and a highly reduced harp. So they look very different from rattling and ancestral wings. And these rattling and purring signals are convergent in function, and that they are both attenuated and manage to attract females, but avoid parasitism from the fly. So essentially, they are both private modes of communication. But we can have convergence at many different biological levels of organization. And investigating at which level convergence occurs in natural populations, whether it's at the phenotype, gene, or mutation level, for instance, can help us better understand the process and predictability of adaptation. And remember that it's rare that we get a window into mechanisms of adaptation on this time scale. Ultimately, what happens now could later be overshadowed by what happens next. So I asked, what is the genomic architecture of the novel wing morphologies, and does it differ between them? I resequenced whole genomes from ancestral rattling and purring individuals and performed 2 GWASs, or genome-wide association studies, on these sequences to identify SNPs, or regions of the genome associated with the rattling and purring phenotypes. Let's look at rattling first. So here's a Manhattan plot with location in the genome along the x-axis. High peaks will represent SNPs that are associated with the rattling phenotype. So we can see multiple peaks across autosomes, chromosomes 5, 10, 11, and 14, that might be associated with rattling, suggesting that it could be polygenic and autosomal. How about purring? I found that purring is associated with multiple SNPs on chromosome 11 and 2 on chromosome 14. And if we zoom in on chromosome 11, we see that the majority of those SNPs are clumped in a gene involved in chitin binding, which is the material that makes up cricket wings. Our GWAS results suggest that SNPs associated with purring and rattling are in similar regions of the genome, but in different genes. And future work should definitely target transcriptomics in these genomic regions, especially during wing development. I have showed you that perhaps there are multiple different pathways at the genetic level to generate novel traits with convergent functions, even on really fast timescales. And the diversification of wing morphologies and songs is ongoing. Our longitudinal tracking over the past 6 years has also uncovered lots of undescribed wing variation, some of which I'm showing here. And for those of you not used to looking at cricket wings, this variation is much greater than what you find across different cricket species. And that makes this explosion of variation an exciting opportunity to investigate and track the genomic underpinnings of the signals that evolution is trying out, some of which will ultimately be successful, thanks in part to relaxed female preferences, and others that may just be intriguing evolutionary dead ends. I would like to thank all of my collaborators and funding sources, and if there's time, I'm excited to take any questions.
And there is time. We've got about 2.5 minutes for questions. So if you do want to ask a question, go ahead and raise your hand while I'm waiting to see if someone does it. Zachary, we'll have you go. Go ahead and unmute yourself.
Hello, great talk. I'm curious if there are any indications that, given the globalization of commerce in the past 50 years or so, these novel phenotypes that have evolved in Hawaii in response to Ormia, are they back spreading into the rest of Polynesia potentially?
Yeah, that is a great question. I will say we have gone back and sampled in Australia and French Polynesia, and it's almost 100% those ancestral types, but sometimes we will find a little weirdo with some of the weird stuff going on. And I'm really curious about checking out or looking into whether that could be like standing genetic variation that was there before and then is being selected on in Hawaii, or that's actually something I hadn't even thought about. Maybe there's backflow through those pathways. That's a really, really cool hypothesis that I hadn't considered.
And then I actually have a bit of a follow-up, but I will ask you to stop sharing your screen at this point.
Absolutely.
So I noticed that with your ancestral population, most of them are not responding to anything. So is it that like you just have a not particularly sexy male call? Is it that females vary over time whether they're responsive? What's going on there?
Yes, and I did not show this, but yes, I arrived in Australia and they were not going to the ancestral loop that I thought they were going to go to. And that ancestral loop is a Hawaii ancestral loop. So those are Hawaiian males. So I think Jay is on this call here, but I was with Jay and together we kind of developed the supernormal stimulus. And it's basically the song just has shorter song intervals. So it's like a much faster version of that ancestral song. And then 90% of Australian females did go to that like supernormal stimulus. Yeah.
All right, that makes sense. Okay, at this point, Matheus, we will have you start sharing your screen. If you have any other questions, you can put them into the Q&A. I'm going to let you know that there is one there already.
Awesome. Thank you.
And then, Matheus, you can start as soon as your stuff is up.
So do you see my screen?
Yes, we do.
Cool. Well, hello everyone. Thank you for being here. My name is Matheus Salles, and I'm currently a postdoc at the Federal University of Paraná, Brazil. And today I'll present part of my PhD research on machine learning-based species limitation. Well, species limitation essentially is the process of identifying independently evolving lineages. Different species concepts and methodological frameworks exist, but in practice, we're always trying to infer meaningful evolutionary hypotheses about boundaries between species. Importantly, species limitation is always hypothesis-based and subject to revision as new evidence become available. Because delimiting species can be so challenging, molecular approaches became increasingly important in this field. In particular, molecular species limitation has been strongly influenced by the multi-species coalescence theory. And approaches based on this theory explicitly model processes such as divergence, ancestral polymorphism, and demographic history. So because of this, they became extremely important in modern phylogenetics. So yeah, of course, these methods also present important limitations. First, they can be computationally expensive, sensitive to model assumptions, and also difficult to apply under specific evolutionary scenarios, like those involving processes such as gene flow. So this motivated increasing interest in alternative approaches, such as machine learning. Machine learning approaches can, for example, efficiently handle large and complex datasets and have already been applied across many areas of evolutionary biology. More recently, they have also started being explored in species delimitation problems. So in one chapter of my thesis, we conducted a review study focusing specifically on machine learning applications in species limitation. And we found that the proposed methods so far are highly diverse, differing in their algorithms, data presentations, analytical strategies, and they include like supervised, unsupervised, and deep learning approaches. Here I'm showing you just a few representative examples from this rapidly growing literature. So one important insight from our review was that species limitation frameworks and machine learning approaches share some important conceptual similarities. So first, in machine learning, methods are broadly divided into supervised and unsupervised learning. And interestingly, species limitation methods can also be grouped into two major categories, namely discovery and validation approaches. And discovery methods aim to identify groups without predefined assumptions, which conceptually resembles unsupervised learning. In contrast, validation approaches will require researchers to first define candidate lineage and then test these hypotheses, making them conceptually similar to supervised learning algorithms. So we propose that machine learning methods may be particularly useful because they can integrate different kinds of patterns while remaining computationally efficient. At the same time, these approaches may also suffer from issues like biased training data or algorithm over-specialization. So careful validation remains essential. Another important conclusion from our view was that no single method obviously is universally optimal. Species limitation depends heavily on the evolutionary context, dataset properties, and model assumptions that are being investigated. We also argue that machine learning may become especially useful when integrating multiple sources of evidence. So we first recommend that when choosing an appropriate machine learning framework, one should consider multiple factors simultaneously, including the evolutionary scenario that is being investigated, the type and quality of the available data, and the assumptions underlying each analytical approach. In some, these new artificial intelligence methods should not be viewed as absolute replacements for traditional delimitation approaches, but rather as complementary tools that may perform well under specific biological scenarios. Well, so far I focused on the broader conceptual landscape, putting that way, of machine learning and species limitation. So moving now from the conceptual discussion to a practical implementation perspective, one factor that immediately caught our attention was the fact that substitution models, like they're fundamental in molecular evolution, their impact on machine learning remains relatively understood. So that caught our attention. And this is important as substitution models parameterize different aspects of sequence evolution. And speaking regarding phylogenetics in general, they influence multiple downstream inferences, like topology estimation and evolutionary parameters. So what we asked ourselves was, how robust are these approaches, these machine learning approaches, across different molecular evolutionary scenarios? At first, we imagined that poorly specified models could potentially affect machine learning performance as well. Conceptually, that there is a trade-off between simpler and more complex substitution models. Simpler models are computationally faster, but biologically less realistic. More complex models better capture molecular evolution, but may increase computational cost and parameter uncertainty. So we wanted to evaluate how much this difference actually matter for machine learning classifiers. And to investigate this, we performed a simulation study using POP AI, which is a Python framework designed for simulation-based population genetic and species limitation analysis. At first, we simulated data sets under 3 substitution models, namely GXCantor, HKY, and GTR. These data sets were simulated under 5 alternative evolutionary scenarios, assuming one of them as the true generating model, each of them comprising the same 3 lineages, A, B, and C. We then trained supervised classifiers separately for each substitution model and evaluated them using independent mixed model test datasets. And yeah, performance was primarily accessed using classification accuracy. Overall, classifier performance was consistently high across all tested conditions. However, one particular interesting result was that performance remained highly stable across substitution models. In practice, classifiers trained under different molecular scenarios still performed remarkably well when evaluated on mixed datasets. Well, what does this mean? First, both biologically and computationally, our results suggest that substitution model differences may contribute relatively little to the broader demographic and delimitation signals captured by these classifiers that we train. We also observed diminishing returns after roughly 800 SNPs, indicating that large matrices produced progressively smaller gains in performance. And overall dataset size appeared to have a stronger effect on classifier accuracy than substitution model complexity itself. But of course, our study still has important limitations. This part of my thesis, we're still developing the preprint version. So far, we did not explore several potential sources of model violation. In addition, it's important to say that correctly identifying models does not necessarily imply accurate estimation of demographic parameters such as migration rates or divergence times. So that would be a nice thing to explore in the future. Also, I think future work should explore more complex evolutionary scenarios, including broader demographic conditions that were present in our simulated models. We also plan to evaluate larger SNP matrices and compare these approaches against another machine learning frameworks that we did not test so far, and another model-based methods. Yes, just to conclude, machine learning first, in the first part of my presentation, just say that machine learning approaches appear to be very promising tools for species limitation, especially because of their scalability and their ability to capture complex evolutionary patterns. But again, careful validation remains essential. In our simulation study, we found that classifier performance was highly robust across substitution models under the scenarios we tested. Dataset size had a stronger effect on performance than substitution model complexity itself. Nevertheless, more realistic evolutionary scenarios still need to be explored before broader conclusions can be made. So yeah, before finishing, I would like to thank the symposium organizers for the opportunity to present my research. I would also like to thank the scientific societies that made both my presentation in this event and my attendance at the in-person meeting in July possible. Many thanks also go to my funders of my postdoc project. And finally, I would like to thank my university and graduate program and my lab, led by my friend and supervisor, Fabricius. So yeah, thank you very much for your attention.
So we've got about 1 minute. If somebody's got a pretty fast question, thank you for stopping sharing your screen. We can get the next person sharing. We do also have a question in the Q&A if no one wants to ask another question in this time period. All right, no one is raising their hand, so I am gonna again direct people towards the Q&A. Matheus, you do already have one question over there. Princeton will have you wait about 30 seconds until we hit the normal start time. As many of you know from teaching, 15 seconds of silence is a lot longer than it feels like it's going to be. And all right, you can begin whenever you're ready.
All right. Hello, everyone. My name is Princeton Vaughn. I'm a fourth-year PhD candidate in Shane Campbell Staten's lab. And today I'm going to be talking about some of my work looking at how lizards are adapting to hurricanes. But we know that the world is changing faster than ever before, and much of this is due to the actions of humans. Wildlife in this current day and age are having to deal with changing climates, urbanization, deforestation, and tons of other anthropogenic pressures. But how does wildlife actually adapt to the novel conditions of a rapidly changing world? Now, this is a fundamental question in evolutionary biology, and to fully understand it, we need to integrate across multiple levels of biological hierarchy. By combining different studies to look across genetics, phenotype, function, and the environment, we can reveal unique information about the process of adaptation that we can't get just by looking at each of these levels in isolation. And to paraphrase George Bartholomew, he says that for any given integrative level, the level above reveals the significance, while the level below reveals the underlying mechanisms. And so to better understand adaptation, we need to look across all of these different levels. And these relationships have classically been represented as a hierarchy, but they're all interrelated and connected. And integrating across these levels requires using lots of different experimental approaches to bridge all these different gaps. And so we can leverage the accelerated rate of global change to help us investigate the evolutionary mechanisms that allow these populations to adapt and to survive in these novel conditions. And natural disasters, such as hurricanes, are actually some of the most extreme examples of rapid environmental change, and so they make for really powerful case studies for addressing these fundamental evolutionary questions. And we know that as climate change continues to progress, these storms are only going to become more frequent and more powerful, making it necessary that we understand their effects on wildlife. And actually, one such study of hurricane selection comes to us from the Turks and Caicos Islands. So researchers were studying two populations of silver key anoles living on two small islands, Pine Cay and Water Cay. And while they were studying them, two very powerful hurricanes hit these islands within the span of 2 weeks of each other. And the researchers wanted to know if these storms had any impact on the lizards that they were studying. But they found that body morphology before the storms was quite different to body morphology afterwards. In particular, they found that femur length, fourth toe length, and snout vent length, which is just body size, all decreased on both islands after the storms, whereas humerus length and toe pad area got larger on both islands after the storms. And from those traits, we can make a series of assumptions about how they play a role in determining an individual's ability to survive the storms by holding on to branches. But no one has really studied the underlying mechanisms or functional consequences of these trait shifts yet. And because these changes appear on both islands, studying these parallel trends can give us lots of insight into how evolution can be repeatable. And this will ultimately help us predict how other populations will respond to hurricanes, especially as climate change continues to progress and these storms become more frequent and more powerful. But so far in the system, we really only know that the environment and phenotype are related. But to fully understand how these lizards have adapted to the storm, we need to traverse this entire circle and make these different connections. So we know that when the storms went through, the phenotypes shifted, but we don't know the mechanism by which that phenotypic change was achieved. And so using genetic data collected from individuals before and after the storms, we can bridge this link between selection and genetics by identifying the genes that are under selection after these extreme events. And because these hurricanes impacted both islands and caused parallel shifts at the phenotypic level, we also expect that the genes under hurricane-induced selection should also be parallel across islands as well. And so we used reduced representation genome sequencing to search for hurricane-accelerated regions of the genome using a method called population branch statistic. And from those data, we pulled out 4 different genes that are outliers in both islands, suggesting that these genes are under parallel selection. Looking at the putative functions of these 4 genes, they all seem to be related to the regulation of skeletal muscular physiology, which makes sense given that we find shifts in limb length in the wild. However, we know that, while there may be a few genes that have a large effect, a multitude of other genes may all interact and compound to significantly influence these patterns as well. So we used whole genome sequencing to search for signatures of polygenic selection. And to do this, we used 2 different complementary approaches. So first, using PC-adapt, we ran a PCA on each population separately. This allows us to detect regions of the genome that are under selection while simultaneously controlling for population structure. Combined with that, we then performed a genotype by environment association analysis to search for genes that are associated with the storm conditions. And it should be at the intersection of these 3 different approaches that we find the genes with the strongest evidence of hurricane-mediated selection. So if we take a look at that overlap between these analyses, we find that over 6,800 genes are in common between these 3 approaches, right? That's a lot, but it's telling us something really important about selection. It tells us that hurricane-induced selection wasn't just limited to a handful of key genes. It was very widespread throughout the genome, indicating that hurricane-induced selection is highly polygenic, really much more than we expect ants. And also within this overlap, we also find those same those 4 parallel outliers in the reduced representation sequencing. So we're only just beginning to explore this expanded data set, but we know that one general drawback of these selection scans on their own is that they can't really tell us if these genes are linked to the phenotypes we know shifted in the wild population. And if a gene is important for hurricane survival, then we expect it to be both under selection and associated with a phenotypic shift. So to statistically link genotype to phenotype, we've performed a series of genome-wide association studies on each of these shifted traits, which allows us to see which of these different loci are associated with each shifted phenotype. And we find that a single SNP within our HS6-ST1 gene, which is one of those 4 parallel outlier genes, has the strongest association with 4th toe length across the entire genome. So this is telling us that HS6-ST1 is both under selection in both populations and has very strong associations with a shifted phenotype. So this all points to HS6-ST1 as being a very promising candidate gene for hurricane adaptation. But again, association alone can really only tell us so much, right? We still don't know whether this gene drives the reduction in toe length that we see in the wild. And so we teamed up with Doug Menke's lab to generate CRISPR knockout anoles to test what would happen if we disrupt this gene's function. And we actually find that the knockout anoles differ in hindfoot morphology compared to the wild-type individuals, with knockouts displaying reduced 4th toe length that was driven primarily by the shortening of that first toe bone. Also, along with that, cartilage levels were reduced in the bones of the foot and of the 4th toe compared to the wild-type individuals. And actually, in fact, one of our mutants displayed complete agenesis of one of their toes. It just never really developed properly. And so it seems that this HS6-ST1 gene is really, really important for building proper hind limbs and proper digits in anole lizards. Okay, so far, we're finding that all the data sort of point to a single direction, right? Toes get shorter after the storms, and a gene that is strongly associated with this phenotype is under parallel selection. But then we can predict that toe length variation, specifically having shorter 4th toes, will lead to increases in hurricane performance ability. But to test this, we're still going to need to connect phenotype to function. And to do that, we had to simulate a hurricane, essentially. So I got this custom-built fan system, which is essentially a hurricane in a box, which lets us achieve really high wind speeds of around 30 meters per second, which is right at the cusp of a Category 1 storm. So we would expose these lizards to these high winds and record each trial. We use Deep Lab Cut, which is an automated tracking software, to digitize the videos so that we could calculate how long each individual is able to hold on to the perch for. Combined with that, we also X-rayed each lizard so that we could identify how morphological variation influences high wind clinging performance. And we find that the bones of the hind limb, specifically the length of the first bone of the fourth toe, significantly impacts performance, where having a smaller fourth toe means that you are able to hold on to the perch for longer. But if you remember back to the video, you might be wondering, excuse me, that that lizard was holding on with its forelimbs. And so why does a hind limb bone actually matter for clinging, right? Well, we actually found that these relationships are hierarchical. So it sort of plays out like this. If a lizard has a short fourth toe, their hind limbs are able to sort of stay on the perch for longer and stay holding onto the perch. When their hind limbs are able to hold on for longer, this reduces the strain on their forelimbs to support the entire weight of the body, which ultimately leads to increases in clinging performance overall. And so it's through this path, shorter fourth toes do lead to increases in hurricane clinging ability, just like we were able to predict from our data. Okay, so to recap, we're finding that this HS6-ST1 gene is under parallel selection on both populations after these extreme events. This gene also has very strong associations with fourth toe length and plays key roles in digit development in anole lizards. When this gene is disrupted, individuals then develop shorter fourth toes. Shorter fourth toes then allows these anoles to better hold on to the perches during the hurricanes. And so taken together, these results indicate that HS6-ST1 is a repeated target of selection, ultimately enhances clinging ability by reducing fourth toe length to better allow these wild populations to survive the storms. But more broadly, this all presents a case study in which we're able to integrate across multiple levels of biological hierarchy, using multiple different lines of evidence to understand how these populations have adapted to the hurricanes. These integrative approaches are going to become even more crucial to use, not only as our climate changes, but as our entire world continues to change in this human-dominated age. And with that, I would like to thank my PI and the other amazing collaborators that we've had on this project. And I'd be happy to take any questions if there's time. Thank you.
And there is plenty of time. So we've got 3 minutes for questions. I will point out you actually already do have one in the Q&A, but let's see if we have anyone else raise their hand and want to ask a question live. Yep, and so we've got one here from Benjamin. Go ahead and unmute yourself.
Hi, great talk. I was wondering if in the knockouts there were any other pleiotropic effects. So you talked a lot about the hind limb morphology, but were there other kind of drastic changes in the phenotype that might suggest any constraints on that gene in general?
Yeah, so we actually didn't notice any, although our analyses were pretty targeted to just the hind limb. But we did measure other phenotypes like body size and other bones of the hind limb. Although we didn't notice any other effects. It was really targeted to those distal hind limb elements that were really being affected by the knockout of this gene.
And I think I will actually read the question from the Q&A out loud so you can answer it live. So Joshua Banta was asking, were these lizards grown in a common garden to evaluate the degree of phenotypic plasticity in their morphological differences?
Yes, that's a great question. So we didn't use the same lizards that were affected by the hurricanes, unfortunately. But what I can say about the sort of weight of plasticity versus evolution are that on these two islands, once the hurricanes went through, this sort of represents a very intense selective pressure on one generation, and so there's like no time for there to be any plasticity, especially in limb length, because we know that limb length is analyzed in anole lizards once they reach sort of adulthood. And so that selective event, like really, is just sort of selection on standing genetic variation, essentially. I can say, if I don't go over time, that the original researchers went back to those same sites a year after those storms passed through, and so like essentially another generation, and found that actually those phenotypic differences remained significant between like the previous generation and one and two generations afterwards.
All right, so that brings us to the end of our session. So I'd like you all to join me in thanking our current set of speakers. We will now have another short break, and there will be another session later on, about half an hour from now, with the remaining set of 6 finalists for the Hamilton Awards.